especially Professor Sumanjan and Professor Abdel for the invitation. Although I am trained and started my career in the area of computational fluid dynamics and heat transfer, but I have started working in microfluidics in last 6-7 years, and I do little bit of microfluidics beside my core areas. In capillary microchannels, so we have already seen from last 2 days that how this cancer cell migrates. So when you have normal cell, the cell division takes place, it grows in order, but when we talk about the cancerous cells, then obviously cell division takes place, but it grows in disorder manner, and you can see that after that these cells from the primary tumor, it actually detaches and goes to the blood vessel and go to the other organs. So that way actually it spreads. So in this work we are actually interested in breast cancer cell. Why? Because when to reach the lungs, this cancer cell has to pass to the pulmonary capillaries, and the pulmonary capillaries are having the dimensions of around 7 microns, 6 to 7 microns. But when we talk about the breast cancer cell, MCF7 or so, those dimensions are of the order of 20 to 30 microns. So the question is that this 20 or 30 micron cells size, how it passes through the pulmonary capillaries which are having 7 micron diameter. And second thing is that if it passes, whether still they are alive or not. So that was our objective, but when we our objectives is to see the behavior of these live cancer cells in capillary like microenvironment and to observe the phenomenon of metastasis. But when we started the work, actually we didn't get this MCF7 from this National Center for this NCCS Pune. So what we got actually HeLa cells, which is not breast cancer cell, it is the cervical cancer cell, but it is having the same dimension of MCF7. So we use these cervical cancer cells, and we cultured in the department of BSB, Bioscience and Bioengineering of IIT Guwahati, and we used these cells for our experiment. So now to mimic these pulmonary capillaries, we actually designed this microchannel. So you can see that this is having the dimensions of 7 micron. This is the constricted passage, and so this is the inlet and outlet, and these are capillaries, but it is then it goes to 7 micron. But so this is the picture taken from the microscope, so you can see here the clear view, because the dimension is 7 micron. So in bare eye it is very difficult to see this channel. So you can imagine that our here size is of the order of 7 micron, and it is 7 micron, so 1/10 of that size. So for fabrication purpose, actually it was very challenging, because that time 6-7 years back, actually there was not much facility in India to fabricate this 7 micron channel. We contacted several private companies, but they told that the minimum dimension they can fabricate 100 micron. So we are fortunate to get the facility at SENSE, Center for this Nanoscience and Engineering at IIT Bangalore, and they extended the help, and we actually got the all these microchannels fabricated there itself in free of cost. So you can see that this is the silicon wafer where we actually these microchannels are printed here. So you can see this is one inlet and outlet, and you can see this is the channel. So in one mask we have almost 30 to 40 channels, and we use this nylon ring and put the PDMS with crosslinker, required crosslinker. So depending on the strength we need, whether it is soft or hard, so depending on this we just use the crosslinker, and we get this imprint in the PDMS, and then once it's solidified, then we actually cut each cake, and we get one such channel where we have one inlet, one outlet, and this constricted channel. So we cut it. So after that, actually we clean, and we actually use the punching tool for inlet and outlet, and this is the open channel is now ready. But now we have to actually cover it, because it is open in one side, so we have to cover it. So for that we use this glass slide. So glass slide, but for pasting we have to use molecular pasting, not any external pasting. For this we use this oxygen plasma chamber, and this pasting is done, and you can see now it is the first channel. So one side we have this glass slide, and other side is PDMS. So we actually see in the microscope from the glass side. So for that you can see that these HeLa cells we have actually got it from National Center for Cell Science, NCCS Pune, and we cultured it in our BSB department. I have one collaborator, Professor Siddhartha Shankar Ghosh. So biological experiment, actually we get help from his student and from his lab, and we use actually DMAM just to separate the cells for using our in our experiments. So for that you can see that these are the procedures, general procedures to actually separate these cells, and we can use it for the microfluidic experiments. So this is our experimental setup. So we can see that this is the syringe pump, and syringe, then these liquids actually with the cells, it comes here, and here we have the channel. It is a inverted microscope, so we visualize from the top bottom. So there the glass is actually bottom side, and it is actually recorded in the high-speed camera here, and actually this is saved in the computer. So with this, now you can see the closed view where we keep the micro channel, here inlet and outlet, and we visualize from the bottom. So with this, you can see that we have done several experiments, and when we see the flow of a single HeLa cell, so it is very interesting. So you can see that diameter of the single cell is higher than the diameter of the constricted passage. So once it comes near to the entry, so it squeezes and goes inside, and it goes out, and again, as it is a single cell, almost the shape is similar but somewhat distorted, and it happens very fast. So you can see from this animation how quickly actually this goes or passes through this channel. So that way it is the visualization of flow of single HeLa cell. But when we get triplet, so you can see that one by one they go inside. These are squeezed and goes and comes out in the other side. But once when it is going, so obviously it is blocking the passage. So this actually velocity becomes very less, and slowly it goes, and it stays for a longer time inside. So residence time is more for the second and third cell. So you can see when the second and third cell it goes, mostly it ruptures. Okay, so cytoplasm comes out as they stays more time in the constricted passage. So you can see from this animation how one by one it actually travels. So once it goes, it blocks, so it waits, then passes. So like that, so you can see that it ruptures. The third cell is ruptured. So now we have cluster of HeLa cells. So it is coming near to the entry region. So one by one they go inside and passes. But now as when one cell is passing, so it is blocking, so velocity is very less. So residence time for other cells is very high. So you can see when it is coming out, actually it ruptures and cytoplasms come out for the later cells which are passing through the this constricted channel. So here you can see from the animation how it passes one by one. So once it goes inside, the other one is going, so one by one actually it passes. But the later cells actually ruptures more. From here you can see. As it is a only single channel, so obviously there is a pressure difference, so one by one it goes. But when we tried with several channels, branches of channels, we have seen that if one is stuck, so it takes the other part of the channels. So now we have now after collecting the cells in the outlet, we have to see that how many cells are live and dead. So for that we have done this fluorescence based study, and we did some this dual staining with this apiridine orange and ethidium bromide. So ethidium bromide actually gives the red color to the dead cell. So with that, so this is the staining procedure we followed, and we use the fluorescence study. So you can see near to the inlet, mostly these are green. So green means we have live cells, where red is your dead cell and orange is your dying cell. So that is not known as a necrotic cell. So in the inlet, so some red cells are there. So those are actually died naturally. And once it is passing through, you can see that when it is we are collecting in the outlet, so you can see more red cells and more these yellow cells. So with this, now we need to count that how many cells are died. So to calculate the viability, so we actually use this trypan blue dye exclusion assay. So actually this we prepare the cells for trypan blue staining, where we use the we wash the cell with PBS, and cells were deeply silenced and collected in a press tube. Then cells were finally suspended in press GMA media so that they can grow again. So we use then some cell counting machine that is known as Countess automated cell counter in the BSB department, and we actually did this study. So you can see that this 10 microliter this trypan blue dye was added in 10 microliter cell, and trypan blue dye is used to stain the only dead cells. So once we put it in the Countess automated cell counter, so it actually counts the total dead cell in the plate. So now we have done several studies. So from the experiment set 1, you can see that first we use the cells at initial condition. Now we use these for two cases. So one is that cells pass through the constricted channel, and we collected and we kept same cells, but we kept in a natural environment, so that naturally they die. Okay, so those are not passed through the constricted channel. So you can see that when we actually initial condition almost viability is 95%, and we keep this sample for the same duration we did the this experiment, let us say 1 hour. So it is 86%, so that those cells actually naturally died. And when we collected the samples at the outlet, you can see almost 50% actually we have this died cell in the samples. So like that we have done experiment 2 and experiment 3, and mostly we have seen that in all cases you can see that almost 50% still we have the live cells which can actually grow and spread the cancer. So although these cancer cells pass through very stresses when it was passing through the constricted passage, but still we have the live cells, and that has the capability to grow again. So for that, before going to that, let us do some hydrodynamic study of this single hammer cell. So we can see that entry region, when it is coming from the inlet, then it is actually passing through the entry region, then it is constricted channel, then it is going out. So these are different regions, and we have calculated the distance travel and how much time it is taken. So you can see that in the entry region, obviously you have as the area is more, so we have the velocity of the order of 7 millimeter per second. But when it goes to this passage region 3 and 4, so sorry, this is 3 and 4, where you have the constricted passage, so it is very fast it goes, so it is 80 millimeter per second for the single cell passing, and in the exit region it is 35 millimeter per second. That is the average velocity for a cell size of the order of 20 micron. So this is the hydrodynamic study we have done, and with that we calculated the deformation index. So when the cell is passing through the constricted passage, obviously depending on the size it elongates. So if size of the cell is more, so it elongates more. So that means the length of the maximum length of the cell when it is passing through the constricted passage, if your cell size increases, obviously L is increasing, so obviously deformation index, which is defined by L by D, so it increases. So we can see that as cell size increases, the deformation index linearly increases. And from there, we have seen the average transit velocity when it is passing through the constricted passage. So we can see that when cell size is very small, so it is very easily passed through the constricted passage, so velocity is high. But when we have cell size higher size, then obviously it takes more time to get enters in the constricted passage, then slowly it passes through the passage, so its average transit velocity is lower. So and when it is actually entering in the constricted passage, so there we calculate the entry time. So obviously for the lower cell size, entry time is very less, but when it is a bigger dimension, so obviously it takes a higher time to enter inside the constricted passage. So now here we have done this again. Cells collected from the outlet, we can see that it is regrown in the cell culture dishes, and after 12 hours, 48 hours and 72 hours, you can see that it started again growing. So that we can observe that whatever live cells are there, they have the capability to grow, although it actually passes through a very high space, and sometimes the cytoplasm came out in the outlet. So from here we can see that we actually answered that when we consider the pulmonary capillaries, where the dimension is very low, but still the higher dimension these cancer cell can pass through the pulmonary capillaries, and again after passing actually it starts growing. So that is the outcome of this our study. So with this, just I will show other study we have done in 1 or 2 slides. So you can see that this is the first single channel we have done. So again we extended this study where we have the EMP cells. This passes through the blood vessel and blood capillaries. Then again MET cells we get. So this we mimicked using the constricted microchannels with these dimensions. You can see that this is the inlet, so it passes and then we have 3 capillaries, and then we collect it at the outlet. So you can see that how these cancer cells passes, and once the one If one channel is blocked, then the other cells passes through the other passages. So here you can see. So these are the other channels. So you can see here somewhat it is blocked, so it is unable to pass. So other cells are passing through the other channels. So we have done some other study for these EMT cells. We have also done some drug uptake on MCF7 cells. Then later, the other student actually worked on these MCF7 cells, where we have actually designed different these capillaries, and we see how when the cell goes and we give the drug, so how actually this mixes with the cells. So you can see here. So cells are going, so you can see here. So here we give the cell inlet, and here we give the drug, and when it passes through these capillaries, it actually mixes. So you can see from here these complicated capillaries. So you can see these are the cells, how it is passing. So this is also having the dimensions of this of the order of 7 micron. But when we actually did this study, so the clean room facility was built at IIT Guwahati. So now our student can actually fabricate these small dimension channels of the 5 micron, 7 micron themselves in our clean room facility at Nanotechnology Center. So all other students actually they are trained in the clean room facility, and they are actually fabricating themselves all these channels. So these are some publications. So Binita Nath actually our joint guided by me and Professor Vishwas. So she did the this first study, and Niraj Kumar Prasad actually did this drug delivery this study. So just acknowledgement. So obviously we Professor Siddhartha Shankar Ghosh is my collaborator from BSB department, and Vinita and Niraj my actually graduated PhD students, and we acknowledge the help we got from the Sense Acid Analyzer. Thank you. Yes.
So good. I mean, very interesting to see that in the multiple channels and how they get separated. So initially you mentioned that if there are multiple cells, then the later cells they take a longer time to pass, and
yes,
that's
so
what
residence time is more, and space they get more. Right. And
so have you tried with increasing the residence time, either by lowering the velocity or increasing the channel length, but with the single cell? That's what will happen.
Yes, but single cell it will be difficult, because once it actually enters, it will pass very fast.
But if I increase the channel length,
yes, that we have not done, but that will be interesting to see that if they have gone through a more space, whether they ruptures or not.
Yeah.
Yeah. That
because here with the residence time also there is a change in pressure, because there is a block.
Yes.
So a
yes.
change in pressure.
Yes.
So I am kind of thinking if we can separate these two effects.
Yes.
And just look at one effect at a time.
Yes, that will be good.
But also depends on the velocity.
Well, also
depends on the
that's why I said either the velocity or the channel
but
length.
some minimum velocity is required to actually pass through this cancer cell, because when it is squeezed and squeezing
Right.
and getting entered in a constricted passage, so it needs some.
You need that pressure to kind
yes.
of push it
Again,
through.
the single cell, that means how much it will be disrupted at the end. That's also.
That's what I'm saying. So that now it is kind of a little bit of multi effect together if we can separate them at. But yeah, that is very interesting.
I think it's very interesting because the mechanical pressure it goes through, how the cancer cell actually going through those capillaries. But if you look at the capillaries, they are very much flexible, right?
Yes.
Unlike the channel you see here.
Yes.
Secondly, there are also endothelial cells, which is not considered here, right?
Yes.
Because we know that there will be an interaction between the cancer cell and the endothelial cells, and the flexibility of the capillaries. None of this has been considered in this case,
Yes.
right? Because that's a real
Yes,
cancer handle.
but we can actually, we are thinking to fabricate the soft, using PDMS, we'll use soft channel, and I'll be interested to see with Professor Pallav also if we can do something that if we can actually put a layer, what you are telling.
Right.
So bioprint if we can do and pass the cancer cell, then how it affects.
Yeah,
so
means-
that will be interesting to
Yeah,
study.
in bioprinting you have a concept of sacrifice thing.
Yes.
You print it and then you put a liquid which will sacrifice that and layer it with endothelial cells, because essentially that will provide you the idea about why the lung is become really the area of metastasis.
Yes.
And
But only thing is that the dimension is very small. So
7
that
micron making that will be
that
actually
with the bioprinting, I do not know, means that
Yeah, that will be
we have to explore. That will
7
that is
micron
challenging.
with the soft material, that will be tricky.
It will collapse.
That will collapse and they will
Or
have.
it will be blocked actually with that.
It will get the cohesion to
Yeah.
kind
Yes,
of
yes.
wall cohesion and well.
So the FSI is two-way or one-way?
No, we have not done the FSI because this is a solid channel.
No, but the body is deforming.
No,
Cell-
body
cell.
here it is not deforming.
The cell you are
Cell
talking
is
of the
deforming.
elongation.
Cell is deforming. Yes,
Ah.
yes.
But that is one way.
That is one way, but it is not a flexible channel.
Another, sorry.
He's having a, he has a,
Ah.
Just a quick question. It's not clear for me, how do you impose the flow? There is a
Yes,
flow.
we use syringe pump.
Okay.
you
So
will-
you impose the flow rate.
Yeah.
But the velocity may be that the velocity of the flow is more, then they are more damaging when they are coming out.
No, so
So
that
have
we are
you
discussing that it maybe say velocity is less, then the residence time will be more. So maybe that time it will
But
rupture.
when the velocity is more, maybe the shear pressure is also more when it is coming out and rusted.
But we have actually considered the velocity, what actually it happens in the blood flow in the
That
micro.
microcapillary,
that
yeah.
is also
that-
true. That is also true what you are mentioning, that velocities come and the collision will be more.
Shear will
Yeah,
be more
so
towards
shear will be
the
more, it will-
wall, and then when it
Yeah.
is coming out, it is rupturing.
It can
Yes,
provide you
yes,
the-
that is also another aspect.
Yeah.
When it is coming out from the S, if
Yes.
it is velocity is more, so it is throwing
Yes.
more velocity. Yeah.
So you just, I may
Okay,
have missed it, it's just to understand the aspect ratio of small capillaries. What is the thickness of the chip? I didn't follow it. Height. The height of the
So
channel.
it is a square channel. So it is of the 7 micron by 7 micron.
So they come from like 100 micron height to 7 micron height, right?
Yes, yes. So
Okay.
if you see this,
So it's not only squeezing one bead, it's squeezing the two dimensions.
yes. So so you can see this is almost 100 micron. It is 60 micron here. So from 60 micron to 7 micron, actually there is a constriction.
Yeah, but the height of the
Height
channel.
is 7 micron.
7 micron all the way through.
Yes, all the way through.
Okay, so
So it is a constant.
they're already squeezing.
Yeah.
Yeah, yes. It is kind of one dimension if you see from this side.
Okay, thank you.
Yes.
Just one comment. Sorry. Professor, did you consider usually if, because you're talking about the resistance time and for the disruption of the, but when the cell, the first cell is passing through squeeze like this, there is also a possibility that the channel surface is also modified because the cell will deposit some kind of other stuff on the PDMS, and now these molecules or other stuff that are deposited on the channel may interact with the following channel cells, and that may also be
So
an effect
it is
for.
a good actually observation. So actually we have used this channel 2-3 times, so we didn't face any problem. So maybe say no deposition is taking place inside the channel.
Time
Because
scale
it is
is
a 7
very
micron
less,
channel.
right? The time scale of the passage
Yeah.
is like, so what is the time scale of the passage? That was like
the passage.
No, I'm the entry to exit. So what is the time?
This is
So-
Few seconds.
So it's very few seconds.
Yeah, but really.
So milliseconds, some milliseconds.
Some milliseconds.
Some
But
milliseconds.
there is one test to do. I mean, you pass cell by cell, okay, and if the
One by one, yes. But
One
it
by
is very
one, and
difficult to
then
control.
there is no effect.
That is very difficult to control because
Yeah.
say we take the cells, all cells, we pass and we observe that when one cell is coming, then we observe when the triplet cells are coming or double cells. So that way. So, but what you are telling that one by one we have to pass. So that is very difficult to control actually in the channel. Yes.
So very interesting, and the way that they are moving through the channels. One aspect here is that the CPCs do not move, most cases, they do not move as singlets.
Yes.
They move as clusters. And it would be interesting to see whether the clusters get disrupted, because even they are surrounded by platelets, like in our wet lab experiments, we incubated them with platelets, CTCs, and also immune cells are there. So it would be very interesting to see how they approach such a scenario of a constriction in a lung. I
Yes.
completely
So
understand.
You can see this paper because this you can go through this paper. So here we have observed this kind of way. So you can see they are actually, this actually passes like in a cluster, right? So you can take here and you can see this paper. So this paper actually, some of the clustered actually when it comes to the near to the channel, so it passes one by one. We have seen. But obviously those are only clustered of in CF7, not other cells.
Also, in continuation to what Shamanta asked, that I completely understand that creating a matrix on the 7 micron would be a really challenging one. But it would be interesting to take those cells after the flow and see whether their attachment to the endothelial layer or endothelial cultured cells have increased or decreased, and that way we can probably have an idea that they have a even if they ruptured, we are just looking into the rupture just after the channel. We probably do not know whether they, because lipid is kind of, they can reassemble, and probably they are able to come back to their original shape, disperse some cells, because cells are known to throughout their cytoplasm if required, and then come back to a particular state. So it would be very interesting to, means for me, I understand from the mechanical point of view the movement. For me, the cells after they have moved are even more interesting, because that probably tells what they have acquired and what they have not acquired, and it will be interesting to check that.
So my comment is kind of, connecting to that, is that after they have moved, you have looked that, the percentage of cells are alive.
Yes.
But it will be interesting to look whether they have the DNA damage response is up, whether the nuclear got blades disrupted.
So I have to collaborate
The
with
mechanical properties after this
After this, the blade formation and
Also with
So
those.
this is a good platform for discussion,
Yeah,
and
well,
then you can carry forward the
This is
further,
the spirit of this workshop, which
That's exactly what we are trying
and we are capable of doing these channels, and my student actually fabricate themselves in the clinic. So they are trained and they become TA, and for next 4-5 years they become TA and they fabricate themselves. So even we also, like Professor Gautam Vishwas, ask for channels, we fabricate here, we give free of cost.
Also,
what exactly what we were mentioning for this workshop, we submitted this proposal.
Okay.
What exactly?
also the commenting on the, what Rajdeep said, recently from CCMB, Professor Santosh Chauhan published a paper in IMBO in a very different context, but they have shown that if a cell got almost completely disrupted, but again they can assemble, and they have given it a phoenix, the name, so they can again reassemble and get into a fully functional cell. So that will be a good thing to check here also.
Okay. Any more questions?
Thank you very much.
Okay.